Welcome to today's episode. In 2009, the Nobel Prize in Physiology or Medicine honored a groundbreaking discovery that changed our understanding of aging and cellular stability. At the center of this achievement stands Elizabeth Helen Blackburn, a scientist whose journey began far from any laboratory, in the quiet coastal town of Hobart in Tasmania, Australia. Born in 1948 to a family of physicians and scientists, Blackburn's childhood was filled with animals, nature, and endless curiosity. She grew up surrounded by 7 siblings, raising tadpoles, collecting jellyfish on the beach, and exploring a garden full of birds, rabbits, and pets of every kind. This early fascination with the living world gradually shaped her passion for biology. From her school days, where she devoured science books and drew inspiration from the story of Marie Curie, to her university years studying biochemistry in Melbourne, Blackburn followed a clear path towards scientific discovery. That path eventually took her to Cambridge, where she trained in the legendary lab of Fred Sanger, and later to the United States, where she made the discovery that would define her career, the structure and function of telomeres. Today, we look at how this remarkable journey led to a Nobel Prize, and how her work continues to influence biology and medicine.
Welcome to For Science Salah Lotfy channel, where global scientific research becomes clear and accessible.
هنا نحول الأبحاث العلمية العالمية إلى معرفة مبسطة مترجمة لك بلغتك.
Ici, la science est expliquée et traduite pour tous.
Ciencia clara, accesible y disponible en varios idiomas.
Wissenschaft, verständlich und mehrsprachig für jeden.
قناة For Science Salah Lotfy channel. العلم للجميع وبجميع اللغات. تابعنا لاكتشاف المزيد.
Welcome to For Science Salah Lotfy channel. In today's episode, we open a remarkable chapter in the history of modern biology, a story where curiosity shaped a scientific revolution and where one woman's journey from the far edges of Australia led to a discovery that changed our understanding of life itself. Elizabeth Helen Blackburn, born in 1948 in the quiet seaside city of Hobart, Tasmania, did not begin her life in a laboratory. Her early world was filled with the rhythms of nature, the sound of waves, the textures of sand and shells, and a home teeming with life. Birds, rabbits, cats, and dogs roamed the family garden. She even kept lizards and tadpoles, nurturing a fascination with how living things grow, adapt, and survive. Her parents, both physicians, created a household where science was not just a profession, it was a daily language. Yet it was curiosity, not obligation, that pushed young Elizabeth deeper. At school, she developed a love for reading and science, inspired by the extraordinary story of Marie Curie. From that moment, the idea that science could reveal the hidden architecture of life became her guiding force. This passion led her to the University of Melbourne, where she immersed herself in biochemistry, and then to Cambridge, a global center of scientific excellence. There, she trained under the legendary Nobel Laureate Fred Sanger, known for transforming DNA research. In this environment of high precision and relentless questioning, Blackburn found not only mentorship, but a new scientific identity. A researcher driven by rigorous thinking and the courage to explore what others overlooked. That courage took her across continents, eventually leading her to the United States, where she embarked on the research that would define her legacy. Working with careful persistence, she uncovered the structure and function of telomeres, the protective caps at the ends of chromosomes that safeguard genetic material. This discovery opened new frontiers in aging research, cancer biology, and cellular stability. It would later earn her, along with Carol Greider and Jack Szostak, the 2009 Nobel Prize in Physiology or Medicine. But the importance of Blackburn's story goes beyond awards. It is a story of how scientific insight grows from curiosity, discipline, and environments that allow questions to evolve into breakthroughs. It is also a reminder that major scientific advances often begin far from the centers of power, sometimes with a child examining creatures on a beach, wondering how life works. In this episode, we explore not only what Elizabeth Blackburn discovered, but how she discovered it. We look at the scientific challenges she faced, the questions that pushed her forward, and the impact of her findings on modern biology and medicine. Welcome to a journey that connects childhood wonder with Nobel-winning science. Welcome to the story of a discovery that redefined our understanding of the cell. and welcome, as always, to For Science Salah Lotfy channel.
University of Melbourne. Early education and the first steps toward telomere science for science. Welcome to For Science, Salah Lotfy channel. Today we explore an important chapter in the scientific journey of Elizabeth Helen Blackburn, the years she spent at the University of Melbourne, and how this period set the stage for her groundbreaking work on the DNA at the ends of eukaryotic chromosomes. Elizabeth Blackburn's story begins far from the major scientific hubs of Europe and North America. She grew up in Hobart, Tasmania, surrounded by nature, animals, and endless curiosity. But her passion for science required a bigger stage, a place where she could move from childhood fascination to real scientific training. That place was the University of Melbourne, one of Australia's oldest and most respected academic institutions. When Blackburn entered the university, she stepped into a world of structured science, a world of laboratories, chemical reactions, and precise measurements. Her chosen field, biochemistry, was still rapidly evolving at the time. Researchers worldwide were racing to understand the molecular language of life, and Melbourne gave her the tools to join that race. During these years, she learned the fundamentals of molecular biology, how DNA replicates, how proteins are built, and how cells maintain their internal balance. More importantly, she developed the disciplined thinking and careful experimental approach that would later define her scientific career. Professors encouraged her to question every assumption, double-check every result, and approach every biological problem with both skepticism and imagination. By the time she completed her undergraduate and postgraduate work, Blackburn had built a solid scientific foundation. But she also felt the pull of bigger questions. The University of Melbourne had given her the knowledge and confidence to pursue research at the molecular frontier. It was here that she first encountered the mysteries of DNA, the molecule that stores life's information, and began to wonder about the parts of the genome that scientists still did not fully understand. This curiosity led her to a fundamental question. What happens at the very ends of chromosomes? These terminal regions were known, but their structure and function remained mysterious. Were they merely leftover fragments? Or did they play a deeper role in protecting genetic information? Melbourne prepared Blackburn to seek answers, but solving such a question required advanced tools and mentorship. That next step took her to Cambridge, where she trained under Nobel Laureate Fred Sanger. But the groundwork, the intellectual discipline, the passion for discovery, and the early exposure to biochemical research all began at the University of Melbourne. Her later work would reveal the structure of telomeres, the protective caps of repetitive DNA that stabilize chromosomes. These structures prevent genetic material from unraveling during replication, and they play a crucial role in aging, cancer, and cellular health. The seeds of this discovery can be traced back to her years in Melbourne, when she first learned to blend theory with experimentation and began shaping the questions that would lead to her Nobel Prize. The University of Melbourne was not just an academic stop in Blackburn's journey. It was the starting line of a scientific career that changed biology forever. It provided the environment where natural curiosity matured into scientific inquiry, and where a student from Tasmania began the path that would uncover one of the most important molecular mechanisms in the cell. In this episode, we highlighted how educational foundations shape major scientific breakthroughs, reminding us that great discoveries often begin with simple questions asked in early classrooms and university labs. Stay tuned as we continue exploring the scientific path that led Elizabeth Blackburn from Melbourne to Cambridge and ultimately to her Nobel Prize. Welcome always to For Science Salah Lotfy channel. Welcome to For Science Salah Lotfy channel. Today's episode takes us into two important chapters in the journey toward understanding telomeres, the earliest attempts to isolate telomere proteins, including the failures that shaped the discovery, and a glimpse of the scientist behind the experiments, Elizabeth Helen Blackburn outside the laboratory. Before telomeres became a central concept in aging, cancer, and genome stability, they were simply a scientific mystery at the ends of eukaryotic chromosomes. Researchers suspected these chromosome ends held something important, but no one knew what they looked like, how they functioned, or what proteins might bind to them. In the early years of her research career, Elizabeth Blackburn faced these unknowns head-on. She had already made significant progress by identifying the unusual repeating sequence at the ends of chromosomes. But uncovering the proteins involved, the molecules that protect, stabilize, and maintain those ends, proved far more difficult. The first attempts were frustrating. When Blackburn and her colleagues tried to extract telomere-binding proteins, the samples often degraded. Experimental conditions that worked perfectly for other DNA protein complexes failed repeatedly here. The ends of chromosomes behaved differently. They were fragile, easily lost, and extremely sensitive to experimental conditions. In some of the earliest attempts, electrophoresis gels showed nothing, just empty lanes where a protein band should have appeared. Other times, the DNA failed to bind anything at all. The simplest hypothesis was that the proteins didn't exist, or that telomeres functioned without specialized protein partners. But this hypothesis did not satisfy Blackburn. Her training, built on careful observation, critical thinking, and double-checking results, pushed her to question the experiment, not the biology. She revisited every step. Was the buffer too harsh? Was the extraction temperature damaging the protein? Were the telomeric sequences too short to attract their natural partners? Most experiments ended in failure, but each failure revealed a small clue. Gradually, she realized that telomere proteins were extremely sensitive and tightly regulated. Isolating them required gentler methods, more precise conditions, and new ways of detecting weak interactions. These setbacks did not slow the progress of telomere research. They shaped it. They taught researchers that telomere biology was built on delicate, specialized molecular interactions. Those early challenges paved the way for later breakthroughs, including the discovery of telomerase and the full understanding of telomere-binding complexes. Every failure was a stepping stone toward identifying the protein machinery that maintains the ends of chromosomes. But scientific discovery does not happen in a vacuum. Behind every experiment, every late-night calculation, and every failed gel is a human being navigating a life beyond the microscope. Elizabeth Blackburn's life outside the laboratory reveals the person behind the Nobel Prize, a scientist shaped not only by data, but by experiences, values, and passions. Growing up in Hobart, she spent her childhood surrounded by the natural world, animals, beaches, gardens, and a home full of curiosity. This grounding in nature stayed with her. Even as her work became more complex and her schedule more demanding, she carried with her a deep love for the simplicity and beauty of living organisms. Outside the lab, she sought balance. She read widely, explored music and art, and made time for her family. She understood that a creative mind needs moments away from equations and samples. These breaks were not escapes but sources of clarity. Moments when ideas settled, reorganized, and sometimes revealed solutions that hours of benchwork could not provide. Her move between continents, from Tasmania to Melbourne, then Cambridge, and later to the United States, added another dimension to her identity. Living in different cultures gave her perspective, resilience, and a broader understanding of how science fits into society. Her life beyond the lab also included teaching and mentoring. She believed that curiosity should be nurtured, not controlled, and she encouraged students to ask bold questions, challenge assumptions, and learn from mistakes, the same approach that guided her early struggles with telomere proteins. These experiences shaped not only her scientific achievements, but the way she viewed discovery itself. For Blackburn, science was never just a series of experiments. It was a lifelong dialogue between the mind and the world around it. Today, we explored how early experimental failures revealed the complexity of telomere biology and how life outside the laboratory shaped the scientist behind these discoveries. Understanding both sides, the scientific challenges and the human experience, gives us a fuller picture of how breakthroughs are born. Throughout every great scientific journey, there are individuals whose presence, ideas, and mentorship quietly shape who we become. For Professor Elizabeth H. Blackburn, winner of the 2009 Nobel Prize in Physiology or Medicine, these influences form an essential backbone to her story, guiding her curiosity, shaping her scientific discipline, and supporting her pursuit of bold questions. People who have had important influences on my life as a scientist growing up in Tasmania, Blackburn was surrounded not only by nature, but also by people who believed deeply in science. Her parents, both physicians, nurtured her fascination with the living world. They never pushed her toward a profession. Instead, they opened doors of curiosity and allowed her to walk through them freely. Their scientific environment taught her early that observing nature was not just a pastime, but a path to understanding life. In school, a few teachers stood out. They recognized her hunger for knowledge, encouraged her reading, and supported her early interest in biochemistry. They planted the idea that science was not a collection of facts, but a way of thinking, a method for approaching mysteries with discipline and imagination. Later, during her university studies in Melbourne, she met professors who shaped her understanding of molecular biology. But perhaps some of the most influential figures emerged during her postgraduate training. Mentors like Fred Sanger at Cambridge, a towering figure in DNA sequencing, and later colleagues and collaborators who pushed her to think deeper about chromosomal stability and the structure of telomeres. Each person left a mark. Some offered guidance, others challenged her assumptions. A few inspired her resilience when experiments failed. Together, they helped build the scientific mindset that would eventually lead her to uncover the role of telomerase and the mechanism by which chromosomes maintain their integrity across divisions. A network of inspiration. But not all influences came from inside the laboratory. Blackburn herself has often pointed to the broader scientific community. Colleagues exchanging ideas over coffee, conferences filled with debate, and students who approached old questions with fresh eyes. These encounters reminded her that science is never a solitary mission. It is a collective pursuit, where breakthroughs rely on both mentorship and collaboration. Even outside the world of research, personal relationships played a role. Supportive friends and family offered her balance, grounding her during the long hours and intellectual demands of laboratory life. Their presence allowed her to return to her work with clarity and renewed energy, maintaining the steady progress required for high-level scientific discovery. Influence as a living legacy. Today, Elizabeth Blackburn's own work has become a source of influence to countless scientists worldwide. Just as she once stood on the shoulders of those who supported her, new generations of researchers now stand on hers. Her legacy is not only the discovery of telomerase, it is the culture of mentorship, curiosity, and integrity that she has modeled throughout her career. Closing message. And as we reflect on her extraordinary journey, we also recognize the profound impact she continues to have, not only through her scientific achievements, but through the example she sets for young researchers. That science advances through perseverance, collaboration, and the courage to ask questions that others overlook. On behalf of myself, Professor Dr. Salah Lotfy, from the Egyptian Atomic Energy Authority, and on behalf of For Science Salah Lotfy channel, we extend our deepest appreciation and gratitude to Professor Dr. Elizabeth H. Blackburn, Nobel Laureate of 2009, for her lifelong dedication to science and the inspiration she continues to offer to the global scientific community. Thank you, Professor Blackburn, from all of us who believe in the power of science to change the world.